everyone. I want to thank you all for joining us today for our ALS Webinar Wednesdays series. My name is Kelly Hall, and I'm a marketing coordinator for ALS North America. Due to a medical issue, Allison Neto will not be hosting our webinar today, but instead we do have our presenter is Lee-Ann Hemphill. Lee-Ann holds a Bachelor of Science degree and diploma in water and wastewater treatment from the University of Manitoba. With over 35 years of experience, she has extensive industry experience in public and private laboratory operations and currently manages food and water microbiology, limnology, and some other industrial hygiene analysis. Now, Lee-Ann serves as the Biology Manager for ALS Environmental in Winnipeg, and today she will be discussing Legionella. During this presentation, feel free to use your chat box, your Ask a Question, or raise your hand feature to ask questions. All questions will be answered at the end of the presentation. Lee-Ann, thank you for taking your time today. I will go ahead and turn it over to you.
Thanks, Kelly, and hello, everyone. Thanks for joining us today. I do want to apologize in advance for any rough patches we might hit with this presentation. I am pinch hitting for my colleague, and I'm going to do my best to present this the way she intended it. Okay, so we're here to talk today about Legionella and try to understand what exactly it is and what the public health risks are. And these are the types of things that have now resulted in the need for monitoring programs or mitigation plans, which is probably why you're registered for this in the first place. We're going to look at a few of the different resources out there available to help you establish risk management plans for your various facilities. We'll talk about some sampling protocols if you choose to use ALS for this, and our sample integrity requirements. We're going to go over the test methods a little bit and then look at what's on the horizon for us in terms of improving our Legionella testing. And again, thanks for your understanding of my situation here. And with that said, here we go. Lime green in color, in other words, they can take on many different appearances, the various species. Some can even fluoresce under UV light, and that is a diagnostic tool that we can use here. And some people describe the classic Legionella bacteria colony as having a ground glass appearance to it. So what else we know about these things? They are opportunistic pathogens with ultimate incubation periods of 10 to 20 days. So it can take quite a long time for us to see this in the lab. That does make it different from other bacteria, which typically grow much quicker. So I guess the message there is this test does take a long time to perform in the lab. And compared to other bacteria, I would say that this one is perhaps not as well understood yet. So we are still learning as we go along. And here's just a picture of a typical Legionella pneumophila colony, a bunch of them actually. This is what it would look like to us in the lab on our media. And this is the species that people generally associate with illness, and it is one of the more easy. This particular slide is pretty pristine. It wouldn't typically be what we'd see from a client sample. This is likely a pure lab culture, and there is no background material on that plate. So it's very nice classic example. So the black background is a little bit different too for us. It's helpful when it comes to looking for these things, but just to touch on why it's black, there is charcoal added to this media. And we know that this is a requirement for growth of Legionella. We don't quite understand how it comes into play in the lab, but it is a requirement. And there are some other specific amino acids required too. And all of this makes it, again, a bit of a challenge for us to understand what's happening. But I guess, you know, to summarize, you would say this is kind of a fussy eater, but it's also a surprisingly robust organism in various environments. And that's what's going to pose a challenge for water systems operators, building operators, in trying to maintain their systems. So just for shock factor, this is the same colony, or one of the colonies, blown up by transmission electron microscopy. We do not do this here. But you can see that it looks very different, and it is a rod-shaped organism. The measurements here, you know, indicate that it is a little bit smaller than other bacteria, and we do have to take some additional steps in order to retain that when we go to process samples. So some other interesting features of this genus is that there are now at least 50 species known, and many more serogroups within those species. We are still discovering, for lack of a better word, new species, and again, lots to learn. Oops, sorry there. This organism can also, it's very hardy, so it can take home in quite a wide temperature range. It does not reproduce below 20 degrees, but it's quite happy up to and including 70 degrees with those specific requirements for growth that we talked about. The other interesting thing is it does like to hang out with other organisms, and so you'll find it sometimes inside the cyst of a protozoa organism. It loves biofilms, again, a big concern for building systems, and it can even live inside human macrophage cells. So it is a little bit of a tricky one to treat at times. All in all, it's got a lot of survival techniques that it employs, and this is part of why it's important to keep your monitoring program current and active. So, some more bad news. It's found worldwide, and it does live happily in the environment. Lakes, rivers, ponds, streams, anywhere there's enough moisture and the proper food sources. What we think now is that these numbers usually are not low enough to create much of a health risk for humans. The problems occur when we concentrate into plumbing systems and facilities. It can remain dormant. I did mention it doesn't like to do much less than 20 degrees, but it can happily subsist there and waiting for better conditions. So this is why you will see populations concentrate in reservoirs like a hot tub or fountains, showers, even respiratory therapy equipment once it hits that preferred temperature range and has the proper conditions to reproduce. Okay, so water temperature, big deal. This table just kind of outlines, you know, very basic thermal treatment options. You can see that the optimum temperature is, you know, pretty close to human body temperature, 35 degrees. Anything below 20 degrees Celsius, 68 Fahrenheit, organism will remain viable, just doesn't do much. In order to kill it, you need to get up above 50 degrees and apply that temperature for an extensive period of time. And that period of time comes down as the temperature can be increased. So this is one effective way to treat a system. However, you know, you're trading one risk for another in some cases. I don't believe you can maintain plumbing systems at 70 degrees if you're going to offer that to the public. So scald risk is another problem. Okay, so we did say that water temperature was very important. Stagnant water is another good indicator that you might start to see some problems. Biofilm formation is a very big favorite, and we know that in plumbing systems you get things like scale forming and sediments. That combination of, you know, inorganic and possibly organic material is a great hiding place for this organism, and it does serve as a food source as well. And if you think about plumbing, you're going to find conditions like that possibly in something like a cooling tower, air conditioning units, and again, all of these other public facilities where there are hot tubs, whirlpools, pretty much anywhere. So where the risk comes in, even though things are growing in there, where the risk comes in is when the physical environments change and this material is disrupted and then released from its biofilm or its happy life underground into the surrounding water and then potentially into an aerosol. So when you have lines that aren't very actively used in a plumbing system, you're going to have that build up, and then when you open up that tap, you release that material into an aerosol form. And obviously, air conditioning and heating towers take a bit of a hit there. It is the most obvious situation where you can have this occur, but it is not the only place, as we've seen in this slide. But with that said, then it also, comes as no surprise that there is a bit of a seasonal pattern to outbreaks of Legionella. So most cases are reported in the months with higher temperatures, rainfall, and humidity. And one example of that from 2013 would be 65% of all the cases in Ontario were reported between June 1st and September 30th. And a quarter of all of those were reported in July. So happy August. Okay, so why are we concerned about this organism? There are two main health implications that can arise from Legionella. One is more serious than the other. Pontiac fever is the lesser known condition that's associated with Legionellosis. It is non-fatal. Symptoms are very similar to many other conditions. And because of that, we feel that it's quite an underreported condition. People put this off as just the flu, and that's that. When it comes to Legionnaire's disease, though, this is the one that, hits the news periodically. It is a type of pneumonia. It can be fatal. And again, immunocompromised groups of people are the most at risk. This is an inhaled condition, and there is no evidence right now of any human-to-human transmissibility. This is strictly an airborne communicable disease. So, to put a bit of a point on it, here are all of the different species that can cause some form of Legionellosis. So we've talked about Legionella pneumophila, serogroup 1. That was the beautiful picture of the white colonies on the black media. Here are some of the others of the 50 to 70 known that may also be a potential source. Okay. One thing we do know, I'll just go back to that slide for a moment. One thing we do know is that a few of these are very frequently recovered from distribution systems. They may be a little less hardy in the lab sense, so we may have a little more difficulty recovering these. So again, we may be underestimating their prevalence in the environment. Okay, so I'll just give you a brief history of a few bigger outbreaks, and you may already be familiar with these. The first one occurred in 1976, so this is again a fairly new genus as far as the world of microbiology goes. Philadelphia, the Legionnaires had a convention, and we all know there was a lot of bad things that happened there. And that is, in fact, how the organism got its name. Another large one in Stafford, England, a few years later. 16% fatality rate would be considered pretty high, I think, by any epidemiologist standards. Again, it was related to air conditioning. Moving forward to the Netherlands, we had a flower show, and about 10% of the people who attended and reported cases did die. This was a large one. This source was identified as a hot tub in the exhibition area. So again, just aerosol generation into a crowded facility, and here we go. Moving into some more recent cases, we had a big one in New York City. This created quite a scene as well, and I do believe it resulted in the state of New York putting in place some of the most stringent protocols out there for dealing with outbreaks after this event. And of course, one that we've heard about in the media recently in the wake of some other related problems was Flint, Michigan. 12 deaths related to this, just adding insult to injury for those people. This seemed to be related to their municipal system and very preventable with proper disinfection. And then most recently in Canada, we did have a little situation in BC with cooling towers. It was not confirmed, in fact, that this came from the cooling tower in Walmart. They did find Legionella in the shopping mall itself. But either way, Walmart voluntarily shut down the store while we dealt with the outbreak. So with all that, the question now becomes, how do you determine when you have a problem that you need to remediate, and how do you find out about your problem? There are various resources out there, and you know, really there is quite a bit online. One of the most important standards, I think, is the ASHRAE standard. That's the American Society of Heating, Refrigeration, and Air Conditioning Engineers. They did put out a guideline. It has been updated in 2018 and is standard 188. They have established some minimum risk management requirements for building water systems. It is designed for use by owners and managers of human-occupied buildings, but it does exclude single-family residential buildings, so not covered in this standard. It's an extremely comprehensive document, and it does go into details about designing your sampling program, and they touch on the types of equipment that might need to be monitored and how to remediate. Anybody who's involved in installing, operating, servicing, or maintaining these buildings, I would really encourage you to buy this. Unfortunately, you do have to purchase it. It's about $100 US. All of this, of course, is still, as we understand it, voluntary monitoring, not mandatory, but in some jurisdictions, I think this has already been incorporated into building codes. And that's probably where this is headed long term. Public Works and Government Services in Canada also has their own standard document for federal facilities. Again, provides the minimum requirements, and this is applied to all federal facilities now. So we do have, you know, contract work going for companies that are maintaining those buildings on behalf of the federal government. They first published this actually in 1986. It has gone through some updates, and you know, most notably, they had to respond to an outbreak in Quebec City in 2012. And you'll note the date on this is 2013, so they did act pretty promptly to update their program. Although this is only enforced for federal buildings right now, it is a good resource for any building operator. And this one is in fact free. So I just want to point out, I'm not going to go into too many details, but it does, they are good resources. They are very detailed on assigning risk levels based on the type of equipment you have, how to design the program. Also talks about what are the acceptable test methods for monitoring, and how and when you need to take action on systems. They will all require, you know, making flow diagrams of your building and understanding the movement of water and where your dead zones might be. And similarly, OSHA in the States, the Occupational Safety and Health Administration, which is part of the US Department of Labor, has a great website, covers every potential situation, and they do have some e-tools available for hospital workers and things like that. They do talk also about sample collection, and they do touch on the personal protective equipment that you might want to employ when you're doing this. So I encourage everybody to go and look at that. This particular slide just gives you an idea of the sorts of action levels that are being recommended right now. So the first action would be just to clean your system immediately, and you can see that the thresholds for taking action are different depending on the type of equipment that you're monitoring. So, you know, a cooling tower with a count of 100 Legionella bacteria, they're asking you to immediately clean and disinfect. That's sort of the end of it. If you get up into the thousands on a cooling tower, then, you know, there's further action they want you to take. And again, these are different depending on which document you go to. So you have to use some judgment here as well. Okay, so these are just some links. Again, by Googling ASHRAE, OSHA, CDC, the Center for Disease Control in the US, or even Health Canada, there's a ton of information that you can tap into there. So I'm not going to dwell on this too much. I can put it up at the end if you like. Okay, if you decide to go ahead and start a monitoring program and you want to use ALS, we can provide you with some containers for that. We do require a sterile 1 liter plastic bottle, and that bottle will come with some neutralizing material in the bottom. We want you to leave it in there. It is meant to take away the effect of any residual disinfectant material that might be lingering in the system. If the water is not treated or not potable, then we're not too concerned about that neutralizing agent. We're more concerned about getting the samples here quickly. We do need the full liter. If it does arrive in a bottle that isn't provided by us, we will put a qualifier on there just to indicate that. So we can't vouch for that container and its sterility. So when you go to collect samples and submit them to the lab, we talk a little bit about sample integrity. And again, this first part of that is to obtain the correct containers. Aseptic technique when you're collecting your samples will also help us to give you the best results possible. Okay, so what do I mean by this? We're just talking about any condition of the sample on receipt at the lab that may have an impact on your final results. So this actually applies to all samples, whether it's bacteria, Legionella, or any chemistry. That can include things like a broken container. We don't want to introduce anything to the sample, and we don't want to lose sample on the way here either. Sample then becomes unrepresentative of the true condition at the time of sampling. Frozen samples, this happens up here in Winnipeg about 6 months of the year at least. Freezing will stress a biological organism and may in fact have the cell burst and die. Obviously, if it dies, it won't grow in the lab, so you may get a false negative result. We don't want that. So we'll reject anything that falls into these categories. When it comes to technique, again, there are some good resources out there, but what we're just asking is that you try to maintain some sterility in the field if possible. Wash your hands, and if you have gloves, please wear them. Disposable gloves are great. Leave that sample container that we send you intact until you're at the point of sampling. There will be a sterility seal on there, and you should not break that until you're ready to collect the sample. We'd like you not to put the sample lid down anywhere. You pick up something from outside the actual target zone. We also ask that you disinfect the faucet head if you're working from a tap itself. Again, I'm not too familiar with sample ports in a place like a cooling tower, whether this is an accessible thing or not. If there's a valve to be opened or taps that you can use, it's helpful to have that disinfected, just so you're, again, representing what's in the water, not what's on the outside of the pipe. And I guess I just need to say that you know, each facility has its own challenges when it comes to sample collection. So, you know, if you're ever in need of advice, we can maybe help out with that if you give us a call. And we do recommend wearing some protective gear if you do think you may have some exposure risk there from an aerosol. We just want the bottle filled. We can accept bottles with headspace. In fact, a little bit of headspace is okay, but we do want it full. And then finally, packing your sample for transport. We just want to insulate against hot and cold extremes. Again, I mentioned our Winnipeg winters can be pretty wicked. So a deep freeze here, you know, sort of means you don't need ice packs in transit, but if you're shipping in the summer, obviously we need to account for ambient temperature. And we do want you to initiate cooling as soon as possible, especially if you're sampling from a hot water port, because we want to prevent the proliferation of non-target organisms. As I mentioned, this one grows slowly. There are many bacteria, and this does live in a little consortium quite often. There are many bacteria that will reproduce even in transit and start to compromise what we can do with it when it gets here. Okay, hold times. The version of the international standard that we're using for analysis recommends that samples be delivered to the lab preferably within 1 day, but not more than 2 days. This right now is the gold standard for Legionella testing, ISO 11731, and it is specified in all of those standards that we talked about earlier. We do want you to protect your samples from sunlight. Now, the bottles we're giving you are opaque, so that's another good reason to use our containers. And I did mention we want them to be cooled, if possible, coming from a hot water tank. Sometimes it's not easy to get your samples here from a remote site within that 2-day hold time. The current version that we are using in the lab of this method does permit us to analyze up to 5 days from the date of sampling. What we do after the 2-day mark is hit, if we are in that 2 to 5 day zone, we will put a qualifier on there explaining this. With this version of ISO 11731, this is acceptable, but I do need to say that there is a new version that we will be implementing, and in that version we lose that 5-day window. So the 2-day hold time will be a thing in the very near future, so that's something to consider. Okay, this is an example of one of our chains of custody. And again, our account managers have these and can put them in your hands. We have electronic versions as well. They're Excel files. And really all we need from you is a little bit of a description here on your sample IDs. Dates and times are very important because of those hold time issues that I mentioned. Account managers can help you with pre-charging this, or as I say, you can take an Excel file and set it up for yourselves. We were looking for information about who to report to and who we're sending your invoice to, how you would like to receive your reports. There really is only one test code that we're interested in right now, and that is Legionella-enum for enumeration -WP, which is a suffix for the Winnipeg lab. These samples can be dropped off at any ALS facility and they will make their way to us, but again, hold times may come into play. Up here on the right, you'll see a priority statement box. We do offer rush service at surcharges. However, for this particular test, there is only so much rushing that can happen. So in this case, because of the long incubation required, the only rush factors we can apply would be relating to getting the results to you after we're finished the test. The other thing I just want to mention here is that when you're filling these out, it's really important for us, especially if this is regulatory work, to understand who you would like to have receive these documents when we're finished. We're only going to be able to discuss your results with someone whose name is on this chain of custody unless we get permission after the fact from you. So the more you can complete this ahead of time and get it in our hands, the better, in good fashion. And again, account managers can certainly help with completing these. So how do we actually conduct this test at the lab? I'm going to try not to get too nerdy here, but I think it's important to understand why things take so long for this particular analysis. So the conventional Legionella method is based on a concentration of your sample in the lab using membrane filtration. So we're literally going to take that 1 liter of water and pass it through a membrane filter and then elute the material off of the filter later on to work with. So we're concentrating it up from the liter down onto a very small surface. We are accredited for this test based on the ISO standard, and we follow it prescriptively, so we do not deviate. The issue with this test, again, is length of time and the need for so much liquid. We need to separate this out into several streams for treatment, and the reason why we need to do that is because, again, it's an organism that likes to live with others, and we need to reduce those background organisms the best way we can to isolate only the target organism, Legionella. So we have to strip this into 3 streams. All of this, in a nutshell, gets put onto agar media, all of it. And we incubate this for 10 days and sometimes longer. So that's the first part of the test. From there, we have to take a look at them and move them on to, again, more selective media to try and reduce the background organism growth and confirm the presence of the target organism. And we take that further if we need to do some identification with using latex agglutination. So you can see that, basically this is a really complex and labor-intensive test and long. So here is just a picture of what membrane filtration apparatus looks like. And you'll notice that there's some, gloved hands here. We are working inside a biosafety cabinet here, and we need to protect our analysts throughout this test from any potential exposure to the aerosols in these samples. So the filter goes on here onto the manifold, and we pass the water through using a vacuum pump. And then we take those membranes onto the media. And I don't know if you recall, but I did mention that this is a smaller organism compared to some other bacteria. So we do use a filter with a typically smaller pore size than we would use for other bacterial testing, just to help recover those organisms. And again, with that, we retain everything bigger as well. So there's where the other treatments come in. We run an awful lot of quality control material with this, and we do need to disinfect this manifold in between each and every sample to prevent cross-contamination. So it's pretty time-consuming. Basically, we're going to hit the sample after we filtered it with heat, because we did mention that heat is something that can kill only above a certain temperature, but all of those other bacteria that might be present are more vulnerable and they will die at lower temperatures. So that's the purpose of that. Acid, same thing. This is a bit of an acid-resistant organism. So once we've applied these 3 treatments, then we've hopefully mitigated the impact of any of those competing colonies and improved our chances of recovering Legionella only. We then take part of that material and spread it out, turn the plates upside down when they've dried out a little bit, and we incubate them at 35 to 37 degrees centigrade. This is put into an incubator with an awful lot of moisture. And if you think about it, that's pretty much what the body is providing for this organism as well. That is body temperature, and of course, you're mostly water, so plenty of humidity. Now we say up to 10 days. Legionella is a slow grower, so we won't see anything immediately, and we just leave those things alone for at least 2 to 3 days. So from there, we start looking at the plates after the 3rd day. We might look at day 3 and 4, maybe day 6 and 7. We are simply making notes about what we see. And there's usually plenty to see. After 10 days, then we are at the end of this first incubation, and we need to make some decisions about which of these colonies that we've seen grow up might potentially be worthy of confirming as Legionella. And I did mention that, all these various species have all sorts of appearances, so it's not an easy task to determine what's what. So here's a great example of a pretty yucky looking plate. This is an actual client sample that was submitted to our lab. You can see that there are the classic looking round white colonies, quite shiny looking. Some of them could be something else like a yeast. And then we have these slimy kind of spreader looking things that may in fact be a type of mold. It could also be Pseudomonas. And what we do know is that Pseudomonas and Legionella really do like to hang out together. They both really do enjoy biofilms, and so it's very likely that that was a Pseudomonas bloom. So again, we're talking about streaking out. Oops, my apologies. Okay, so for the confirmation step, we then go back to those plates with all the funny looking colonies and pick them off of there literally and re-streak them onto more selective media. One of these media provides all the food for Legionella colonies. The other is deficient in their requirements. So what we are looking to see is these same colonies grow on one set of plates, but not on the other. And that's a very good indication for us that we've got Legionella. There are a few exceptions to that rule where, again, it becomes really important for the analysts to be well trained and understand how to distinguish what all this means. A lot of interpretation required. And that's where we have, we're very fortunate. We have some really experienced staff here and call upon them daily. So going forward, this is again, here's a charcoal-based media. You can see that there are definitely patterns of growth here, but the similar colonies that were streaked onto this red media, which actually contains some blood, is growing nothing. So we're going to take these guys and say, yep, they need to go ahead with confirmation. I should point out that in that last slide that you saw, there are no gloves on this employee. This isn't from our lab, so that's naughty. Okay, so to get to the species level, what do? We have latex agglutination kits. There are 3 sets of reagents. I'll show you a picture of this briefly. We're looking for a response to the reagent with the antigen for the serogroup that we're possibly going to find in the sample. There's a picture of these things, and you can see here you have clumping in this central zone. So what that tells us is we have a Legionella pneumophila serogroup 2 to 14 happening here. If this had agglutinated over here, it would be serogroup 1, and over here on circle 3, we would consider that to be any one of the other species that we talked about. Okay, so there's your picture of that. I should mention that the new version of the ISO standard does not require us to identify Legionella to species. So that will be a change coming. Now, detection limits. Right now, we are using fairly conservative detection limits for the 2 groups. We will report to you as Legionella pneumophila, and we will identify the serogroup, or all other Legionella species. We will, if there's nothing there, you will get a report that says less than our detection limit for those 2 groups, always for the 2 groups. There is a situation that can occur where we have growth that does not meet the detection level. So for instance, Legionella pneumophila with a detection level of 10, we may see 4 colonies. You're going to have a report that says less than 10, but we are going to report to you that we did find that organism in quantities less than our detection limit. So you'll never be in a situation where something grew on a plate and you didn't know it was Legionella. So I did mention that this requirement to not talk about species is coming out of the version that we are going to be implementing soon in the lab. But with that said, we are not going to take away the opportunity to know the species if you want to know it. We do have here something called a riboprinter, and we can take that species ID forward using that equipment and actually be maybe a little more specific than these latex agglutination kits are now. Of course, it comes with a cost, so if you're doing source tracking or something, this might come into play. Again, that's a good question for an account manager later. Okay, so what else are we doing? There's been a lot of talk about qPCR. PCR stands for polymerase chain reactions. This is a pretty standard technique now in most labs. Right now we use it here in Winnipeg for bacterial pathogens in food. But we do have a plan to bring this in for Legionella. This is a separate ISO standard. It's standard 12869. Again, another prescriptive test. So we are in the process of getting that ready for a release. So really briefly, polymerase chain reaction uses the organism's DNA. It's a much more rapid test. However, it does come with some limitations. And the biggest one is that we can't tell you whether the material we retrieve is living or dead. It's just present. The other thing that I can tell you about this that is a big con is that the methods is very prone to matrix interferences. So for building systems where there are lots of disinfection chemicals involved, it makes it very difficult to get reliable results. Quite often you'll get no result. So this is another reason why we're taking our time bringing it forward, is we've found a company that provides kits that can apparently mitigate that problem, and we'd like to validate that. Okay, so the good news is it's not a 10-day test. We don't need to grow the organisms up. We just simply start out the same way with your sample. We filter it in the lab, and then we take the DNA right from that filtered portion. There's a little bit of a process that goes on, and then we're measuring Legionella directly in the instrument. So the good news is it's a very quick test. As I said, we don't need to confirm anything. The result coming off of the instrument is considered confirmed. It's highly specific, so there is almost no chance of a false positive. It takes away the subjectivity of the analyst. Again, the training issues that come in with being able to recognize what you need to confirm. And it is very well recognized around the world. So AFNOR, I believe, is a European standard, and these two standards work well together, so it's good for a global company like ours to be taking on something like this. Here's what a PCR unit looks like. As I say, right now we're just using it for food pathogens, but our hope is to have this available for client use for Legionella in the next little while. Okay, we talked a little bit. I'm not going to go too technical here, but we're looking at a very specific part of the genome of Legionella. It's very highly preserved, so it's not subject to mutations. It's easy to recognize. And the PCR works on a three-phase system. First, we take the material and amplify it, and then put it through the instrument with the optical detectors. There's a camera involved, and we quantify it over time. And you'll get a result that's expressed as genomic units per liter of sample. Most of the guidelines are looking for 1 genomic unit now. But again, that doesn't mean much unless you know it's alive. Okay, very quickly, here is a DNA ladder up at the top of the slide. So if you can picture a ladder, this thing is normally twisted. The way this works is we split that right down the middle, top to bottom, and then take those rungs of the ladder. There's a section of that is very specific. As I say, we are going to attach our primers to that by using heating and cooling, and then the instrument will replicate that many, many times, and that's how we grow up very small amounts of DNA into large enough amounts to detect. So just to back up here, again, we're working on this method. It's got advantages, and it is written into some of these standards as a good remediation monitoring tool, because you can have your results right away. So in a case where you've had an outbreak and you've gone in and done some work and need to go back in and resample, you can have an answer fairly quickly this way about how effective your disinfection process was. So this is kind of where we end it in terms of what goes on at the lab end of things. I hope that we've given you a little bit of insight into why this test takes so long, typically, and why we can't really speed it up. I'm just going to now take a moment for some shameless promotion of our lab here, because we are unique, and we're kind of proud of that. So on this slide here, you will see, some of the capabilities that we have that other labs do not. Of course, I got biology up here at the top because it is the biggest biology lab we have, I believe, in Canada for sure. I'm not sure about North America, but We also run things like Cryptosporidium and Giardia here, which you won't find anywhere else. And I did mention our ribo printer. On the limnology side, we've got the very traditional environmental limnology work. So we look at invasive species, do a lot of microscopy work, big into algae and bioassays for effluent regulation testing. We also have a small industrial hygiene group, and we look at industrial mold from various sites. We look at mold in air, and we also have a large asbestos lab and radon. Over on the chemistry side, you'll see some specialized organic work, HAAs for sure, and we even offer chlorophyll testing, which is a nice complement to our limnology work. So there, I've done it. We have an expanding food lab, and that too is unique, I think, for you ALS. Okay, I'm done with that. I just want to thank you all again. I'm going to put up some contacts here. So this is me. Barb is our lab manager in Winnipeg, and Wa takes care of the chemistry side. And you know, we'd love to hear from you if you do have any work you want to do. And if you do need any more information about Legionella testing with ALS, please go ahead and give us a call or contact your local ALS, and we'll hopefully be able to help you out. So I just want to thank everybody again for hanging in there and being part of this today. And I guess if we have any questions, I've got an account manager here with me as well, Judy Dahlmeyer. She's our senior account manager here in Winnipeg, and hopefully we can answer your questions. So thank you again.
Yes, thank you, Lee-Ann. We will go ahead and open it up for questions. Remember, you can use your chat box, you can use your ask a question box, or even the raise your hand feature if you want to ask Lee-Ann or Judy a question directly. I will give everyone a few moments. We already had some questions come in throughout the presentation, so we'll go ahead and ask those in 1 second. I just do want to remind everyone that this webinar has been recorded, and all participants of this webinar will receive a follow-up email once the recording is available to view. And we do post all of our webinars on our ALS Global website and our YouTube channel. There you will find a wide range of video resources and also past Webinar Wednesday presentations. So I'll go ahead and start with some of the questions that have already come in. And there is a hand raised. I will get to that one in 1 second. The question was from Bruce. He asks, are all species dangerous or just some? You mentioned that they exist throughout the environment, but are the ones more prevalent in the environment perhaps less dangerous compared to that which grows in the warm water environment?
So what we know so far is that tends to be true. The natural environment does not seem to be a good reservoir for concentrating these organisms up. We are still learning an awful lot though, and as I say, they continue to find new species all the time, so I'm going to use caution when I answer that. Right now, the belief is that the populations just are too low in the general environment to cause an infection. So if you were swimming, you know, swimming in a pond or a lake, this wouldn't be something that would be of a concern. And yes, there are so many that are now being associated with Legionellosis that, you know, we do have to put some cautionary words around that statement.
Thank you. We have another one from Gordon. He asks, it is a legal requirement in Ontario and Quebec currently, but not in any other provinces yet.
Thank you. We know that Quebec has their own set of rules, and yeah, this seems to be very piecemeal right now by jurisdiction, how far into this they want to get. So was the question, does Ontario have a legal requirement, or was that the question?
It's sort of just, yes, that was just the question. Is it?
Yeah.
Yeah. Also,
I can't answer. We're here in Manitoba. I'm not aware of any formal requirements in any specific province to do this, other than federal buildings, which, you know, it's the government. Yeah, yeah. So far, we haven't seen any hard regulations come in. But please, if you're out there and you know of something, correct me, because as I say, it seems to be very patchwork at the moment.
We have another question from Christopher. He asks, could you please confirm the detection limit? Is it 10 CFU per milliliter?
So in our lab right now, that is what we're using, is 10 CFU per milliliter for Legionella pneumophila of any serogroup. And for the other Legionella species, we work at 100 CFU per milliliter, simply because, again, there are so many unknowns and so many novel species starting to come out that, you know, the level of confidence in saying that we absolutely can ID everything on a plate, you know, definitively as either Legionella or not. I mean, we're leaving ourselves a little bit of room there. And this is why we do report out anything that we take to confirmation, whether that detection limit has been met or not. So with the new version of this same method, the detection limit will be 1, provided that we can filter an entire liter of water. So the detection limit will change with the volume of water filtered once we implement that new method. And there are cases where we just cannot push a liter through. So then your detection limit would raise based on that. But our intention is to get to 1.
We had an additional question from Christopher. He asks, could you please speak to swab samples and how the lab processing might be different from bulk water samples?
Oh, yes, sure can. Right, so there is something that didn't make its way into this presentation. Thanks for asking. With a swab, I guess there would be many times when all you have to access is a small port, or you have a deposit that you'd like to collect by swab rather than water. And we have had this happen recently, actually, evaporating trays on systems and things. Okay, so the good news is we can also provide you with the swabs. When they come to the lab, these are directly straight onto the media. And I wish I had someone from the lab here to describe that, but we skip the filtration step, obviously, because there isn't one. So we take it straight onto the media, and then from there the method is very similar.
We had another question come in from Eleanor. With regards to sample collection, what if a whole liter cannot be collected?
We will take whatever you can give us, and again, we will, you know, qualify these results a little bit. We're reporting on waters, we're reporting per milliliter. So, you know, we can do some math and just be aware that, you know, a full liter was not processed. But again, the results, the units of measure are per milliliter of water. So we do have a little bit of room there. That on that same note, then, when the swabs are reported, we do, I believe, report per swab.
Thank you.
We will know the area swabbed. Yeah.
We do have an hand being raised. So Merritt, if you did want to ask your question, you now are able to speak at this time. Merritt, did you want to ask your question to Lee-Ann?
The question was answered. Thanks.
Oh, perfect. All right. I don't see any additional questions coming in. So again, I just wanted to thank Lee-Ann and Judy for being here today and for this presentation, especially with these circumstances. And thank you to everyone who attended today. And hopefully everyone can enjoy the rest of their week. Thank you.
Thanks.